First of all, I would like to thank the organizers and specifically Pierre-André, former colleague of mine at the College of Agriculture of Rennes, who I didn't met for maybe 20, 25 years. So it's been a real pleasure to be here today in Rome. Okay, so now we are coming close to the conclusion of today, and you've got a huge amount of information on butyrate. So it's quite a challenge to try to add on this outstanding presentation. I will try. I will reduce my talk from many slides, I'm afraid, but it's the way it is, okay? So initially, I planned to give you some information on butyrate and gut barrier function, because this is a key function in keeping healthy not only the gut, but the entire organism. The second point, and I will really develop this one, is a link between butyrate and a very important intestinal enzyme, which is called intestinal alkaline phosphatase. It's a very potent anti-inflammatory enzyme, and has also links with the gut microbiota, gut pro-inflammatory components, etc. The third aspect was about butyrate and gut-inducible heat shock proteins, and I would guess that this could add to the discussions one group had earlier this afternoon on heat stress in poultry and even in other species. And fourth point on butyrate and epigenetics, but just as a flavor, because this is already a big story, and the time is too short for doing this. So I can skip this, three major functions of the gut, so physiology, microbiology, immunology. Then the kind of messy situation at the gut interface, I would say, between the luminal contents with food, nutrients, dietary components the organism cannot digest, the microbiota, and a number of metabolites and even constitutive components of the microbiota. Then the epithelium layer, very, very major to protect the organism and the gut. Underneath the lamina propria with all these components of the immune system, and then the smooth muscle layer, so with a combination also of enteric nerves. So we've seen with the previous speakers the links between the gut and the brain. So I can skip this. All of you know this by heart. I can skip this one as well. Because there are microbiologists in the room that know this better than myself, but there are a few key butyrate producers that one should remember. Okay, so given the very many functions, I had planned to focus on barrier functions. I will go very fast, and then on proliferation and link with this enzyme and heat shock proteins. So I skip this one. So for the gut barrier function, I just made a very quick review of recent literature on this, because say 10 years ago there were not very many data. I will not comment this in detail. The previous speaker did a lot on it. I just want to draw your attention on that these effects have been shown in different types of gut epithelial cells of different species and origins, which means that this is probably a kind of universal mechanism. Okay, so I will skip this one. And then moving to the intestinal alkaline phosphatase story. So first of all, so a group in Chicago, now more than 15 years ago, showed that among the short-chain fatty acids, butyrate was the strongest in inducing this enzyme, intestinal alkaline phosphatase, so AIP. And so why is this enzyme so important? So this is a kind of summary of many of its roles. It's a figure I made in the first review I did on this topic 9 years ago, nearly 10 years ago. And since then, there have been many additional information showing the strong protective effects of this enzyme. And I've published two other reviews on this topic, one in 2014 and another one that was accepted last December that should be released early. But you can see, I will not comment all the functions of this enzyme, but you can see that it can contribute to regulate energy through fatty acid absorption through the small intestine. It regulates surface pH at the epithelial lumen surface. But the major points are its, I would say, fantastic role in being able to detoxify pro-inflammatory bacterial components. Here, when I made this picture, there was only data on LPS, but since then there have been data on also flagellin and other nuclear components from bacteria. On the right-hand side, so it's more or less corollary of this is also its ability to regulate intestinal bacterial translocation, which is another story, but it has been shown. And now this enzyme has profound anti-inflammatory effects, not only at the gut level, but also at the systemic level. And part of it is related to the fact that this enzyme is produced at the apical side of the enterocyte, but also at the basolateral side. I will show it in more detail. So I'm talking of the intestinal isoform of this enzyme. There are different isoforms. One is a tissue non-specific isoform, which is found nearly everywhere. But this one is produced exclusively by the enterocyte, so mostly at the apical brush border, and part of it down also at the basolateral level. In the large intestine, normally the expression and activity of this enzyme are very low, and when they are high, it's quite often synonymous with inflammation. So when one looks at the small versus large intestine in interpreting the data, it's very important to know which isoforms have been produced, not to make misinterpretation. And then at the plasma level or serum level, usually high plasma total alkaline phosphatase activity is synonymous of inflammation, even generalized inflammation, and might be considered as a marker of inflammation. So coming back to the enterocyte, so this enzyme is mostly produced by the cell, exported at the apical side, and released through hydrolysis of the anchor part, released in the lumen. Part of it is produced or released at the basolateral membrane, so probably explaining the small contribution of this isoform to the total plasma or serum alkaline phosphatase. It's low, it's below 10% of the total activity. The interesting point here is that this enzyme, when released in the small intestine, will go down to the large intestine and will resist partly to digestion, which means that it's still active in the colon. In terms of mechanisms, it's an enzyme that acts with through a very simple chemical reaction. It's a dephosphorylation of a number of moieties like LPS. And once these LPS molecules are dephosphorylated, they are no longer recognized by their receptor, like TLR4 receptor for LPS. So this breaks the inflammatory cascade. But at the same time, at the enterocyte level, this enzyme is able by itself to inhibit various steps of this inflammatory cascade. I will not go in further details. You can find the details on the slide. Beside this dephosphorylating and detoxifying activity of LPS, I said flagellin, CpG, DNA motif, but also non-bacterial phosphorylated moieties or molecules, like, for example, extracellular ATP, ADP, AMP, and these are called or known as danger signals, very strong inducers of immune responses. So this enzyme is also able to dephosphorylate these moieties to go down to the adenosine molecule, which is, by essence, I would say, anti-inflammatory. This is kind of summary. I have no time to detail everything. You can go to my reviews, but it's a summary I published as an editorial in the American Journal of Physiology some years ago, showing that AAP, by acting on one side LPS, flagellin, etc., and on the other side extracellular ATP, is able to modulate the composition of the gut microbiota. And I would say, unfortunately, so it's strongly anti-inflammatory. That's the big message to remember. Unfortunately, when inflammation is going up, a number of inflammatory cytokines are inhibitors of the expression of the gene of fat enzyme. So inflammation is really key in the landscape. And then it's also an enzyme that connects with the immune system. And for example, it has been shown to inhibit tissue infiltration by neutrophils, this time in fish, which means that, again, it's a kind of a universal enzyme that has a fundamental role in the body. So moving now to gut inducible heat shock proteins. So these heat shock proteins are highly conserved proteins, so like intestinal alkaline phosphatase, with a wide variation in molecular weights between 10 and more than 100 kilodalton, depending on the class of HSPs. And many of these HSPs are constitutive, so they are found in the cells, and their concentration for a given cells, excluding epithelial cells, do not vary a lot. There are molecular chaperones involved in protein folding and also membrane translocation of transcellular membrane, etc. But one unique feature of epithelia, and specifically the gut epithelium, is that there are some classes of HSPs that are inducible. They can be inducible under different condition of stress, like heat stress, because we had the question earlier this afternoon. But they can also be induced, I would say, preventively at the advantage of the organism, for example, with glutamine. And this can help to help animals to go through critical periods, whatever, pigs, birds, probably humans, etc. So in this case, this inducible form of HSPs contribute to protein stabilization and prevent them from cellular denaturation and degradation. Okay, I showed only two examples of this inducible heat shock protein. On the left, HSP27 or 25, depending on the host species, which is an enzyme which is key in the stabilization and reinforcement, I would say, of the tight junctions. I will not go again deeper in tight junctions, but these HSPs, for the smaller molecular weight one, act at the tight junction to strengthen the gut barrier. On the right-hand side, it's to show that the other class of inducible HSP, HSP70, is involved so in cell protein trafficking and interferes with misfolded protein to prevent them from proteasome degradation in kind of emergency situations. Okay, and it also has a strong anti-apoptotic effects. So now, are these HSPs are induced by secreted molecules and metabolites of bacterial origin? I will comment only the frame part in red. In fact, normal butyrate is one of the short-chain fatty acids that are really strong inducers of HSPs. This is shown in different types of cells. I will not comment into detail, but most of these are intestinal epithelial cells. Against non-epithelial cells do not react to butyrate, for example, by inducing these HSPs. So sometimes, depending on the type of cells and the conditions of challenge with butyrate of a short-chain fatty acid, either only one HSP or both classes of HSP would be induced. What do we know on the sensors and cell signaling pathways involved in bacteria, or in this case, short-chain fatty acid and butyrate induced HSP induction? In fact, we don't know a lot of things. I found only one paper back in 2007, but this was not intestinal cells. It was human breast cancer cells showing that GPR receptors were involved, so in this case GPR 43 and the MAP kinase pathways also. So I would guess that this holds true for the Enterocyte, but this has to be shown. Okay, so in summary, the gut microbiota and probably particular bacterial species in strain are responsible for gut epithelial protection mediated by inducible heat shock proteins, and butyrate is a major bacterial metabolite stimulating inducible HSPs in the gut. So as part of the answer to the question about heat stress, I think those who are involved in this business, please look at HSPs. It could contribute to explain differences between treatments. So now regarding butyrate and epigenetics, I will not go very deep in this story, just to remind you, but it has already been said that so epigenetics involve different modifications like histone acetylation with the acetyl group coming from acetyl coenzyme A, and deacetylation through histone deacetylase. Okay, and in fact, as been told, the butyrate is an inhibitor of this enzyme, which increases the access to the genes and their expression. One important point is the second part of the slide on the left lower side is these so-called epigenetic marks can be transferred transgenerationally. And this type of question, early programming and long-term effects, was raised in our group of discussion earlier this afternoon. So this, I think, there is space for experiments and for hypotheses in this area. I will not detail this, just to say that in many studies we saw with the previous speaker that histone H3 is quite often a target for butyrate action. And okay, so again, making the link between butyrate and epigenetics, so of the gene for intestinal alkaline phosphatase. It's been shown in the early 2000s, still by the group in Chicago, that histone acetylation is involved in the overexpression of this enzyme, and again it links to butyrate. I came to this story of intestinal alkaline phosphatase precisely with findings coming from early programming experiment in rats. So the idea was to see how the gut or some gut function would be affected with this, because 10 years ago most of the experiments and knowledge were related, for example, to the first to the pancreas, and second, and so glucose regulation, etc., and second to the brain. But at that time there was nothing on the gut, and by doing I would say rather a simple experiment in rats, we could show that this enzyme was the only one of the gut we tested. We tested nearly 10 enzymes. That was the only one that was imprinted that the early environment of the offspring looked at the adult age. Maybe I have no time to go into details. If you have questions, I can explain. But the idea to retain is that there was an imprinting in this case. And when we crossed this early conditioning and late or adult life nutrition, having two treatments, normal fat diets versus high fat diets, we could show that with a well-balanced diet, there was no difference in the enzyme activity between the two groups of offspring. But when they moved to, or part of them moved to high-fat diet, the one have been treated in a special way in utero and during early infancy through the mother's diet, was not able to adapt it to increase the level of his enzyme in order to control inflammation. Is said in short, okay? And more or less in the pig with another model, a model of providing antibiotics to the sow in a window around farrowing, we went with this enzyme more or less to the same story. Not exactly the same, because one can imagine that there would be interspecies differences, of course, but the basic idea is that there are a number of fundamental functions that can be programmed very early in life, even in utero, and with maternal influence, but also with paternal influence, which are far less studied. So coming to the conclusion, I will not add too much on top of this. So I would say to answer the question, yes, the butyrate makes part of this complex landscape at the gut level and is involved not only in providing energy to the system, but also in stimulating defense systems, whatever they are, I would say, either targeting, either reinforcing the host defenses or targeting a number of toxic compounds from the gut microbiota. Okay, thank you.